We start off with the germ cell. This germ cell must undergo meiosis in order to produce sex cells. When a male sperm cell is created, it is called spermatogenesis. When a female egg cell is created, it is called oogenesis. Meiosis takes place in the testes of males and the ovaries of females. Meiosis is preceded by interphase, where DNA replicates, creating an exact copy of each chromosome. Germ cell lines are diploid, but after undergoing meiosis, they create haploid cells. The number of chromosomes in a parent cell is different than those in a sperm or egg cell because a sperm or egg cell must only have the chromosomes needed. The other half comes from the other cell. Meiosis 1 begins with prophase 1. Replicated chromosomes become visible and the sister chromosomes become joined at their centromeres. Next, the nuclear envelope starts to disintegrate and the spindle fibers start to form from the centrioles. Homologous chromosomes start to line up next to each other. While this happens, an event that is unique to mitosis occurs. During this event, the two homologous chromosomes participate in a crossing over of DNA between homologous chromatids. After this event, the sister chromosomes are no longer identical. We now have reached metaphase 1. In this stage, the homologous chromosomes line up along the equator of the cell. Once the chromosomes are lined up, the ends of the spindle fibers attach to each centromere. In the next stage, anaphase 1, the spindle fibers recede and pull the chromosomes, still double-stranded, to opposite ends of the cell. When this occurs, homologous chromosomes are separated into different cells, so that each cell will have a complete copy of the DNA. In telophase 1, the spindle fibers disintegrate and cytokinesis begins. Two cells have now been created, with the two daughter cells having half the amount of chromosomes as the original cell. After cytokinesis is complete and the cell is fully split, meiosis 2 occurs. This division is identical to that of mitosis, and the daughter cells created by this division have the same number of chromosomes as the cell at the end of meiosis. In prophase 2, the spindle fibers once again start to form at the poles of the cell from the centrioles. Once we have reached metaphase 2, the chromosomes line up along the equator of the cell. The difference in metaphase 2 from metaphase 1 is that there is only one of each homologous chromosome lined up to the equator of the cell. After this, anaphase 2 occurs. During anaphase 2, the sister chromatids that are lined up in the center of the cell move in opposite directions along the spindle fibers. Next, in telophase 2 in cytokinesis, the cell splits and now new nuclear envelopes are formed around the DNA material. We now have 4 genetically different haploid sex cells.